Well, I've been happily asked to introduce Megan, who I was just reminded has been rescheduled because she was turned down for her seminar by Mother Nature. It was a snow day, like really our one and only snow day this year, and I'm glad to announce that. Megan did her bachelor's degree up on Mount Olympus at SFU. And then she came down to do her PhD here at UBC in genetics. And then I guess she went and she decided to slum it as a postdoc. Well, she went to Milan to do that at the San Rafael Scientific Institute. I wouldn't mind doing a postdoc in Milan. That was probably pretty nice, wasn't it? Now she is professor of surgery here at UBC and this also with the School of Biomedical Engineering. She has a list of accolades that are as long as my arm. She belongs to many elite committees, many committees also that I'm sure that she would prefer not to be on, but has been invited to participate in because of the committees that she enjoys participating in. And boy, she's got some beautiful publications as well. You should read them. I'm not going to delay this introduction any further, but she's going to tell us about engineering Tregs for tolerance induction. Megan, thank you so much for joining us today.
Thanks for the intro. Thanks everybody for coming out. And it was actually just as well that had been canceled by Mother Nature because I'd accidentally double booked myself to give 2 talks on that day, one for you guys and one for the University of Alberta. So it was much more relaxed to just do the U of A one on that day and then to come out here separately and do this one. So I work on regulatory T cells. Apologies, it's going to be fairly hardcore immunology for the audience, but hopefully I'll manage to take you through some of the basic concepts and you can catch the big picture even if you're not a deep immunologist. Before I start, just want to highlight that I do have patents and a license related to the A2 CAR that I'm going to be talking quite a lot about today. So my lab works on this problem of trying to control immune responses in the context of transplantation and autoimmunity. And as I'm sure most of you know, the only way that we can make transplant a feasible therapy these days is by using broad spectrum, very potent immunosuppressive drugs, which do mostly stop the immune response against the transplant, but also have a lot of terrible side effects and put patients at high risk for cancer and infections because they suppress the immune responses in general. And then autoimmune diseases, similarly, the only treatments we have these days are very non-specific, broad immunosuppressive agents. And so the holy grail is to try and figure out how you could more specifically only target the undesirable immune responses and leave other parts of the immune system functioning normally. So the way we think the one approach we could do this by is using what we call adoptive cell therapy with regulatory T cells. And the overall concept is to take regulatory T cells either from the patient themselves or from another source, which would be called an allogeneic source, and to put them through some sort of in vitro expansion step in the lab. And this serves 2 purposes. One is to significantly increase the number of cells because the idea is you want to infuse a lot of these regulatory cells so that you tip the balance more towards immune regulation and away from immune effector function. And secondly, because if you put them in the lab, of course, you're now introducing the possibility to genetically modify the cells, to introduce DNA or remove DNA that then helps you to have a more desirable effect. So how is it that infusing large numbers of this one type of immune cell, CD4 positive regulatory T cells, could have such a potent effect and do something like specifically control the response to a kidney or some other sort of transplant? And the reason why we think these cells are better than a conventional immunosuppressive drug is that they have many different mechanisms of action. So unlike a single drug that's just inhibiting one pathway or stimulating one pathway, Tregs are living cells. They respond to the local environment And they themselves then express many different secreted proteins, cell surface proteins that at the local and microenvironmental level can control all sorts of different types of responses. So Tregs have many different mechanisms of action that have been described over the years, but I just want to highlight probably the most important ones. They're listed here. And it's really all about controlling the interaction between the regulatory T cell and the antigen presenting cell. So some of the things Tregs do, and I'll have one more slide on this, is they express very high levels of an inhibitory receptor called CTLA4. And this will actually physically bind to co-stimulatory molecules on antigen presenting cells and remove them, thereby rendering those cells less able to go off and stimulate other T cells. They also make TGF beta, which is a very well-known immunosuppressive cytokine. They express high levels of the high affinity IL-2 receptor, yet they don't make their own IL-2, meaning they consume IL-2 from the environment, preventing other T cells from having access to that cytokine. And then they also express CD39, which is an enzyme that ultimately results in the production of adenosine, which is also another immunosuppressive molecule. And so by reshaping the local environment, by having all these different mechanisms active, what regulatory T cells do is they can actually control the activity of other T cells in the environment that may or may not have the same specificity as the Tregs. And furthermore, they can convince some of those T cells to turn themselves into new regulatory T cells through a process called infectious tolerance. So the idea then is that if you give a bolus of regulatory T cells once, that those cells don't actually need to be there, persist in the long run, because it would be these newly generated recipient-derived Tregs that go on to maintain tolerance lifelong in the patient. That's the dream, at least. So just a little more detail on this mechanism of action, because it may be one that some of you haven't heard of before. So most of you have probably heard of the CD28 co-stimulatory system. It's the classic thing that we teach you in Immunology 100, basically. T cells express CD28. They bind to these two proteins on antigen-presenting cells, CD80 and CD86, and normally that would deliver a positive signal to T cells. But there's another receptor called CTLA4 that binds to these same two proteins, but with a much, much higher affinity. And so if both CD28 and CTLA4 are present, CTLA4 will win out. And what Tregs do is they express very high levels of CTLA4, and through a process called transendocytosis, will actually physically remove CD80 and CD86, bring it into their cell, and through a degradation process, actually destroy the protein CD80 and 86. And so overall, your antigen-presenting cells become stripped of this co-stimulation, and that's one of the main ways that Tregs will suppress immune responses. So Tregs are T cells. Like all T cells, they need to be activated by a T cell receptor. So all those mechanisms I just went over get upregulated when the cell encounters its peptide MHC molecule. But I'm sure many of you know that there's different ways now that you can use engineering to control the activity of T cells. And in particular, we've been spending a lot of time working on controlling T cell activity using chimeric antigen receptors, which has been used a lot in the context of CAR T cell therapy for cancer. And just in contrast to a T cell receptor, a CAR is like a cross between an antibody and a T cell receptor. So on the extracellular side, there's a single chain antibody fused with a linker. This provides the antigen recognition. And then on the intracellular side, it's quite modular. So you can take domains from different receptors that you're interested in. But most commonly for T cells and Tregs, we'd be using domains from co-stimulatory molecules like CD28 and then CD3 zeta, which is one of the key signaling domains of the T cell receptor. And when we look at whether you want to engineer a T cell or a Treg with a T cell receptor or a chimeric antigen receptor, there's pros and cons. They both have therapeutic applications. But if we just focus on engineering Tregs with CARs, some of the advantages over TCRs is that because they're built on antibodies, the antigen binding domain can be made to be very high affinity and also lower cross-reactivity, because TCRs, we now know, tend to be quite cross-reactive. They bind to many peptides. CARs are independent from any endogenous T cell receptor. One of the limitations of CARs is that they can only bind to things that are on the outside of a cell. So like an antibody, the antigen needs to be exposed, so either on the surface of a cell or at least in the local environment. And that's different from a T cell receptor, which can bind to intracellular antigens presented on peptide MHC. And then in the cancer world, usually people talk about CARs being independent of MHC molecules as being an advantage. But since we work on transplantation, actually all the CARs that we work on are MHC restricted by purpose. So I just want to give you a couple slides of background of work that's been published a few years ago. And then I'm going to tell you about 3 vignettes of recently published and ongoing work in the lab. So a lot of the work that we've done has been focused on a CAR that's specific for an MHC class 1 protein in humans called HLA-A2. It's very commonly mismatched in organ transplantation because there's no intentional MHC matching in organ transplant. And this classic second generation CAR with a CD28 co-stim domain and CD3 zeta signaling domain has been used by our lab and other labs around the world to test whether or not this hypothesis that you could express this A2 specific CAR in Tregs and then get them to control a patient's immune response against an HLA-A2 positive organ, whether this was actually a promising therapeutic strategy. And I wouldn't be here talking to you about it if it wasn't. So yes, it is. And some of the ways that we tested this concept, I'm just going to highlight here. So one of the initial experiments we did was to ask how the expression of the CAR controlled homing of regulatory T cells. So we did this experiment where we put an HLA-A2 positive skin graft onto a mouse and then side by side put an HLA-A2 negative graft, and then injected that mouse with our CAR Tregs that had also been transduced with luciferase, so we could track where the Tregs are going using bioluminescent imaging. And what we found in comparison to mice that received control Tregs, so didn't have a CAR specific for HLA-A2, so the Tregs here are in blue, that they basically disappear very rapidly. They don't survive, and they certainly don't go to the graft. So the A2 positive graft is the one in the dotted circle here. Whereas the mice that got the A2 CAR Tregs, you can see that they traffic very rapidly only to the antigen-bearing graft. So they're ignoring this side-by-side A2 negative graft. And they stay there and they divide and ultimately exit to a local draining lymph node. So these data showed that you can really control trafficking when you engineer in specificity. This experiment was all done with human cells. In order to ask more mechanistic questions, we've also set up the system to work in fully syngeneic mouse models. So in this case, we're taking a wild type Black6 mouse, putting on a skin graft that's HLA-A2 positive, and then putting in mouse Tregs that either have the control or the A2 specific CAR, and then asking how long the skin graft survival is extended. And you can see pretty clearly only the mice that got this single injection of A2 CAR Tregs have a prolongation of skin allograft survival, whereas the antigen non-specific Tregs basically have no effect. So I will, however, note that this is certainly not long-term tolerance. These grafts are definitely rejected. Skin transplantation is a really stringent, it's the most stringent model of transplantation. And in the absence of any conditioning, no T cell depletion, no other immunosuppression, it's not that surprising. We didn't get long-term tolerance. And I have a lot of people in the lab now working on how to get this red line that goes down to basically stay flat. And I think that we'll get there eventually, but there's a lot of different things to test. But nevertheless, this work and work from other labs has already led to clinical testing of this approach into ongoing clinical trials. So this first one is using one of the CARs that we created here in my lab. And the idea is to have people who are undergoing kidney transplantation. They'll have their CAR Tregs manufactured in advance and then infused about 100 days after the organ transplant. And the hope is that the people who get these cells can then have their immunosuppression gradually tapered in order to move towards the goal of diminishing the long-term need for immunosuppression and transplantation. And then a second clinical trial with a similar concept being done in liver transplantation. So it's probably going to be 3 or 4 years before we know the outcomes of these studies, but it's exciting that they're already ongoing. But nevertheless, as a basic science lab, we just have so many more questions about how CAR Tregs are working, how we can use them in different contexts. And so that's what I want to focus my talk on today. Some work trying to study the structure-function relationship between CARs and Tregs. And then I'm going to show you some new data where we've been trying to study the biology of CAR Tregs in the context of islet transplantation. And then I'm going to end moving away from transplantation and tell you about a new CAR that we recently made that we think is relevant in the context of type 1 diabetes. So what's the best structure for a CAR in Treg? So when we first started working on CARs, we really were not super smart. We just copied what the cancer people had done, which was use this classic second generation CAR with a CD28 transmembrane and intracellular co-receptor domain, and that clearly worked. But we needed to know whether that was indeed the best structure for Tregs. And so we set off on an ambitious project to test all these different possible co-stimulatory domains. The top ones all coming from CD28 family members and the bottom ones coming from TNF receptor family members. And we were especially interested in these bottom ones because in the cancer world, some of the clinically approved CARs actually contain domains from, for example, 41BB. It's one of the most common clinical CARs. And so we thought that these TNF receptors could be very useful in Tregs. So the way we tested this was we set up a humanized mouse model of graft versus host disease. It's a bit of a contrived model, but with human cells, it's what we could do. So these mice are injected with A2 positive human PBMCs in the presence or absence of Tregs expressing one of those different CAR constructs. And these are just the controls. So we had known from our previous study that CAR Tregs expressing this CD28 version here in blue would extend survival and minimize graft versus host disease, whereas Tregs expressing an irrelevant CAR or expressing a first generation CAR here with no co-stim domain basically are not effective. And then we got a surprising result, actually, that all these other versions of CD28 family members really were nowhere near as good as this original CD28 containing CAR. Even a single point mutation here in red was enough to make these cells less efficient. And possibly even more surprising was that when we tested the CARs with the TNF receptor family domains, they actually were worse than not having a CAR in there at all. So for example, this TNF receptor containing CAR, the mice die faster than if we hadn't given them any Tregs. So this was quite surprising because it was the opposite of what we would have expected from the cancer literature. I'm not going to go into all the details, but we basically ended up finding that the ability of a CAR to work or not really depended on how it allowed those Tregs to interact with antigen presenting cells. And in here, we were specifically studying dendritic cells. So for any of you who may have worked on Tregs, the classic way you test their function is with a T cell suppression assay, where you look at the ability of your Tregs here to suppress your conventional T cells. And when we looked at how well our different CAR Treg variants suppressed T cells, we saw there was no correlation between our in vitro data and what happened in vivo. And so I'll just take you through one example here, which is the 41BB expressing CAR here. So this CAR works very well in vitro. So the 41BB CAR Tregs are very good at suppressing CD8 T cell proliferation, but they're very bad at suppressing graft versus host disease. So poor correlation in this T cell suppression assay. But on the other hand, what we found is there was a very good correlation when we looked at this CTLA-4 dependent mechanism. So we're looking at the ability of the CAR Tregs to remove CD80 and 86 from the dendritic cells. And here, if we, I think it's not labeled the 41BB, but let's look at the TNF R2 here. You can see we have a very good correlation. So this CAR here is bad at suppressing graft versus host disease and also bad at suppressing CD80 and CD86 expression. So these data suggested that actually we'd been looking at the wrong thing. We shouldn't be looking at how these cells interact with T cells. We needed to be looking at how they interacted with antigen presenting cells. So that was interesting, but one of the main caveats of this model is that APCs in this humanized mouse model really engraft very poorly. Obviously, these mice are extremely immunodeficient. There's all sorts of caveats to trying to study normal immune responses in them. So once we knew that we thought it was an APC-dependent mechanism, then we had to go back to our mouse system and basically ask the question all over again, to try and understand in a fully complete immune system what these CARs were doing and whether we would get the same result when we were working in a mouse that had a full complement of antigen presenting cells. So Isaac in my lab went off to basically do it all over again. He made a slightly smaller subset of CARs and set off to re-ask this question: which CAR is better for Tregs, CD28 family or TNF receptor families, as listed here. And when we first did our experiments in vitro, so here just using a very reductionist model where we have our CAR Tregs, and we're looking at the ability of these cells to be stimulated to proliferate and get activated in response to HLA-A2. We got very similar results to what we had seen with the human study, in the sense that this original CD28 domain containing CAR was clearly the best. So it stimulated the most proliferation in comparison to all the other CARs we had tested. So these were basically exactly what we had seen in human cells. And when we went and looked at a T cell suppression assay, so looking at the ability of Tregs to suppress transgenic T cells that were specific for ova, again, the CD28 CAR in blue was the most potent. So it looked like, okay, in the mouse model, the CD28 is giving us the same result. But then when Isaac went in vivo, he got a different result. And actually, he found that all of the CD28 domain containing CAR Tregs, 28 or ICOS or even PD1, which is an inhibitory receptor, all of these CAR Tregs could delay skin allograft survival, which was different from what we'd seen in the humanized mouse model. But however, when we looked at the 41BB CARs, we did find a similar result where the GITR, OX40, and 41BB they were all less good than the CD28 containing CARs than we had in this mouse model. So suggesting that some similarities and some differences compared to the results in the human system. So in order to try and make sense of this, we decided to go back one step, which was to completely remove any co-stimulatory domain and go back and test a first generation CAR. So any of you who've been working on CARs in the cancer field may know that the cancer people started off initially using first generation CARs and found that they were not effective, probably because tumor cells usually don't express co-stimulation. And so the T cells really needed those extra signals from the CARs. And so we had basically skipped over this construct for all of our work and gone straight to this construct. But what Isaac found was for Tregs, actually the first generation CAR works just as well as the second generation CAR in this skin allograft model. So here the red and the blue lines are basically on top of each other. That's suppression of skin allograft rejection, and then this is suppression of donor specific antibodies, HLA-A2 specific antibodies. So what's going on here? How can we reconcile what we're finding with Tregs and what we had found in our human and our mouse studies? So if you go back and you think about how Tregs are naturally activated, so they naturally would have a T cell receptor, and they would express various levels of CD28 and CTLA4 and be stimulated by an antigen presenting cell. And I told you that at the beginning that one of the core functions of Tregs is to shape the function of antigen presenting cells. And antigen presenting cells, of course, express a lot of their own CD80 and CD86. In our humanized mouse model, we have a deficit in antigen presenting cells. And so in all likelihood, the Tregs were receiving a signal through A2, but they weren't receiving a signal through co-stimulation. And so they needed that co-stimulation in the CAR itself. But in a fully immunocompetent mouse model, we now have normal antigen presenting cells again. And so potentially the reason why a first generation CAR can work is that they're now receiving the natural co-stimulation through the antigen presenting cells. And in contrast to a tumor, where a tumor cell may not be able to deliver those signals to a T cell, Tregs have the expressed purpose of interacting with an antigen presenting cell where they could then receive these signals from a natural source. So that was our hypothesis that Isaac went off to prove. And indeed, we think that this is exactly what's happening. So because we're targeting Tregs to APCs, they don't need endogenous co-stimulation because they can get it through the natural source. So the way Isaac did this was he did some reductionist experiments where he had an artificial antigen presenting cell that either did or did not express co-stimulation. So in the absence of co-stimulation, the CD86 negative A2 positive antigen presenting cells are our second generation CD28 CAR is very good at stimulating Treg activation. And the other CARs are less good, although for some reason the 4-1BB is working okay in this assay. But as soon as you put CD86 on those APCs, you can see all the different CARs now just come back to the same level. So basically showing that the Tregs are integrating the signal they get from the natural co-stim with the signal they get from the CAR, putting them together to result in activation. So this was in a reductionist model. We also wanted to ask this using real dendritic cells. So here's an experiment where we're looking at the ability of Tregs to suppress 80 and 86 on dendritic cells. And it's the raw data here, and just focus on the average data here. So the first generation CAR is in red. Yes, is in red. And if you if you don't block CD80 and 86 and you let them just get their natural signal, both the second generation CAR and the first generation CAR Tregs are equally functional. But if you block this signal, in this case by putting in a recombinant form of CTLA4 Ig, you completely block the ability of these first generation CAR Tregs to work. So showing that they really are responding to this natural signal. This is in an in vitro assay. And we could show that this was also true in vivo, where if we injected our mice with gave them a skin graft and injected them with the different types of Tregs, and then gated on dendritic cells, either coming from a graft draining lymph node or from the spleen, what we could see in the draining lymph node is that both the second generation and the first generation CAR Tregs are functional, so they're able to suppress CD80 and 86 to the same degree. But in the spleen, which is probably not a relevant place for the response to the transplant to be happening, basically there's no effect. So these data show that really in vivo, the first gen and second gen CARs are equally able to control dendritic cell activity. So these data collectively have taught us that Tregs definitely need a signal from CD28. This is different from conventional T cells that can quite happily get their signal from 4-1BB or other co-stim molecules. But this signal can be delivered either via the CAR or via their native receptor. And as a consequence, first generation CARs may actually be sufficient if you're going to redirect your Tregs to interact with an antigen presenting cell. And in all of the work that we've been doing now, it's not the effect of Tregs on T cells, but rather the effect on antigen presenting cells that most predicts what the outcome of our in vivo experiments are going to be. And just as an interesting aside, so in both our mouse and human system, Tregs are clearly very negatively affected by TNF receptor family members, at least in the format of the CAR. And this actually is consistent with findings from the cancer world, where CD19 CAR T cells that encode 4-1BB have been shown to result in less undesirable expansion of Tregs and better patient outcomes. And so one of the reasons why in cancer these CARs might be better is that they don't promote Treg function, whereas the CD28 ones do. Okay, so I want to move on now to islet transplant. And in particular, talk to you about this idea of infectious tolerance. So if you remember back to one of my very first slides, I said the fundamental premise of Treg therapy is that those cells should be able to turn other T cells into new Tregs. So this process called infectious tolerance was coined more than 20 years ago, but nobody has really gone back to test whether this truly happens with engineered T cells. And in order for this therapy to make sense at all, it really has to be true. So we thought we had better start tackling this idea to understand whether our engineered Tregs are capable of inducing infectious tolerance. And layered on top of that is the concept of bystander suppression, where Tregs can suppress T cells with distinct specificities. Because if you want to control immunity to an allograft, that allograft has many different MHC mismatches, many different minor antigens. It's not just one thing. You need to spread tolerance to a whole bunch of different antigens. So Tregs with a single specificity have to be able to control other distinct types of specificity, and that's called bystander suppression. So we thought that islet transplant would be a good setting to ask these questions because we have both alloreactivity and autoreactivity. So when you give islets back to somebody who has type 1 diabetes, they still have their autoreactive cells that are specific for insulin and other islet-derived proteins floating around in their body. And it's been very clearly shown that if you give someone an islet graft, those autoreactive cells come back, and together with the alloreactive cells can contribute to allograft rejection. So the question we wanted to ask was whether if we put in Tregs that could control these alloreactive cells, would they also be able to control the autoreactive cells? And so this was basically the idea. So we have our A2 CAR Tregs. We know they can control the A2 positive reactive cells, but are they able to control the diabetogenic autoreactive T cells? And the way we did this experiment, I'm just going to go through a few slides on the model because it's kind of a slightly different model of islet transplantation that we were lucky to set up thanks to Majid Mojibian, who's now in my lab, but used to be in Tim Kiefer's lab and set up this model when he was working with Tim. And it's actually to transplant the islets in the anterior chamber of the eye. And it's got a lot of advantages over the traditional kidney capsule model. Here's some beautiful islets in the eye. The insulin producing cells are red and the glucagon are green. So they go into the eye and they are just nice and happy. And the eye is permissive for both autoimmunity and, as I'll show you in the next slide, alloimmunity. So here's an experiment that was done in NOD mice, so mice that are going to get type 1 diabetes. Before they have diabetes, here's an islet graft put into their eye. So they have nice red insulin producing islets. But as soon as those mice become diabetic, they destroy all the islets in their pancreas, all the red is gone, and they also destroy all the islets in the eye. So autoreactive cells can traffic to the eye and destroy beta cells. And the eye is also permissive for allo rejection. And we've been studying this by using some human islet transplants, where together with Tim Kiefer's lab, we've been putting either normal human islets or stem cell derived islets into the eyes of mice. And in this case, injecting pathogenic HLA-A2 specific CAR T cells, which will very swiftly reject the HLA-A2 positive islets. So these are human islets in the eye of a mouse, happily making human C-peptide. The islets that are A2 positive, as soon as you inject at day 0 the A2 CAR effector T cells, they're very swiftly rejected. And then the other super cool thing about the eye is that it's a window into what's going on because the eye is transparent. So you can put the live mouse under the microscope and watch what's happening. And in this case, together with Francis Lin, we're putting in islets that are GFP expressing under control of the insulin promoter. So each one of these little green dots here is a little cluster of islets in the mouse eye. And if you don't give the mouse any T cells, those islets live happily ever after. But if you give the A2 CAR effector T cells, they're very swiftly rejected. So this is just showing how permissive this site is for studying allo and autoimmunity. Okay, so back to our question. So we knew that A2 CAR Tregs can control alloreactive cells, and we wanted to know if they can also control autoreactive cells that would have a completely different specificity. And the way we did this was by taking an immunodeficient mouse and transplanting in the eye islets that were HLA-A2 positive coming from pre-diabetic NOD mice. One of the little asides that we weren't expecting is that because these islets are coming from mice that are going to get diabetes, the islets actually came with passenger lymphocytes. So the T cells that were already going to the islet getting ready to cause autoimmunity. And those cells engraft into these mice. So these CD8 and CD4 T cells are passenger autoreactive cells coming along with the islet graft here. But what we actually intended to do was intentionally inject these diabetogenic TCR transgenic T cells specific for an islet antigen they're called BDC T cells. The question was whether A2 CAR Tregs could control diabetes induced by these T cells. This is just some data showing the phenotype of the A2 CAR Tregs going into the mice. So they express nice high levels of the CAR, and we have a conjugate marker, and they express our two favorite transcription factors, Helios and FOXP3, which are defining the Treg lineage. So what happens? So the mice that don't get any Tregs in blue here, they very rapidly get diabetes. So after about 20 days, they're all have to be euthanized. The mice that get polyclonal Tregs also are largely not protected, maybe a couple of mice hanging on here, but you can see the A2 CAR Treg recipient mice are almost all protected from type 1 diabetes, suggesting that these Tregs have actually mediated bystander suppression and have suppressed these BDC T cells that have a distinct specificity. So there's lots of interesting things in this model, one of which is that these mice have two different sources of insulin. They have their native pancreas and they have the islets that we put in their eye. So we can start to look histologically at what's going on and where is the protection happening in these mice. So in the mice that get the polyclonal Tregs, in the eye, you can see the islet graft is sloughing off here, looking very unhealthy, whereas the mice that got the A2 CAR Tregs have a nice preserved graft. And this tolerance here in the eye is spreading over to the native pancreas, because if we look in the islets of the pancreas, the mice that receive the A2 CAR Tregs, they also have nice islets that are protected from autoimmunity, whereas the polyclonal Treg treated mice, their islets are basically destroyed. So these data show that the A2 CAR Tregs are protecting both the endogenous and the exogenous transplanted islets. Using the conjugate marker, we can track what happens to the Tregs. So this is looking at engraftment of the cells in blood. So they do engraft very well, whereas the polyclonal Tregs, they don't have an antigen. They don't tend to stick around for that long, but the CAR Tregs are there. And then we can look at the effect of those cells, both on the passenger lymphocytes that came with the islet, as well as our intentionally injected BDC T cells. So if we look at the number of CD8 T cells in the blood, the A2 CAR Tregs have essentially completely prevented engraftment of those cells, whereas the polyclonal Tregs, or the mice that just got the BDC T cells, there's pretty much no effect. Those cells engraft. And similarly, the mice that get the A2 CAR Tregs, they suppress the engraftment of the BDC transgenic T cells, whereas the polyclonal, or the ones that didn't get any Tregs, those BDC T cells are taking off and causing diabetes in the mice. And so overall, if you look at how we've managed to reset that tolerogenic balance, if you graph the ratio of BDC T cells to Tregs, whereas in mice that just got BDC T cells or just got polyclonal Tregs, you have many more BDCs than Tregs. In mice that got the CAR Tregs, we've completely switched that balance now so that we have a more even ratio, almost equal numbers of the A2 CAR Tregs and the BDC T cells. So we've managed to achieve this goal of resetting that balance between effector cells and autoimmunity. So of course, the most important question is what happened to those BDC T cells? They're suppressed, but did they respond to these infectious tolerance signals and turn into Tregs? And I think overall the answer is yes. We're still in the process of doing experiments and analyzing all the data, but if we go in and we look at expression of FOXP3, which is a Treg lineage defining transcription factor in the BDC T cells, which were completely sorted to be negative for FOXP3 before they went into the mice, you can see that over time, with every weekly bleed, more and more of these BDC T cells are turning on FOXP3. So suggesting that really the Tregs have not only mediated bystander suppression by preventing the cells from inducing autoimmunity, they've also mediated bystander or infectious tolerance by turning on FOXP3 in these cells and turning at least a portion of them into Tregs. And we kind of know as long as you have about 10% Tregs in any T cell population, then you'll have good control of immune responses. So in conclusion for this middle part of the talk, we're quite excited to have shown that our alloantigen specific CAR Tregs can also control autoimmunity And this is really the first evidence that infectious tolerance can happen with engineered Tregs. And I think it's going to have a lot of important implications for the whole field, because this phenomenon really needs to work if to make sense. And then we have a lot of ongoing work, some of it summarized here. So does the effect really require an islet allograft? Can we give the antigen in a different place? Is there something still special about the eye environment? And we have lots of questions to ask about tolerance, and of course, transitioning to do the work in fully immunocompetent mice, because there's still lots of caveats to doing things in the immunodeficient setting. Okay, so in the last few minutes I have left, I want to tell you about a third vignette. Honing, so I started in transplant and then I went autoimmunity plus transplant. Now we're going straight into autoimmunity with no transplant, which is trying to make CARs for type 1 diabetes. And when we first started playing with CAR Tregs, of course, we immediately thought diabetes would be a great place to use these cells. But when I went to talk to my diabetes friends, Bruce and Francis and Tim, I said, what would the target be? They didn't really give me any great ideas. And so we knew that we needed an antigen that was membrane bound. We knew it had to probably be specific for the islets, but also present in the local draining lymph node, because if Tregs are working through antigen presenting cells, they need to be stimulated in lymph nodes, not only in the graft. And so when we scratched our heads and were wondering what we were going to target, not coming up with many good ideas, until actually I was in a seminar in this exact room where an invited speaker, Brian Fife, was talking about how he was making antibodies against peptide MHC molecules to study antigen presentation in diabetes. And we suddenly realized that this would actually be a really good approach to making a CAR. So it's a slightly mind-bending immunology here. So just to remind everybody, a T cell receptor normally binds to a peptide MHC molecule on an antigen presenting cell. A CAR can bind to whatever you want, but it has to be on the surface of the cell. It doesn't need to be MHC. But what we're talking about now is making an antibody that has a TCR-like ability to bind to a peptide MHC molecule. So we're still targeting that same thing that we would have targeted with a T cell receptor, but we're using a CAR. And so you might ask, well, why would you use a CAR? Why don't you just use a T cell receptor? And the answer goes back to the fact that you can really have super high affinity interactions if you use antibodies, and you can also really engineer the intracellular domains of CARs in ways that are not really currently feasible with T cell receptors. So there are advantages of using these TCR-like CARs. So we set up a collaboration with Brian and his person in his lab, Justin. So they had basically had made these antibodies that bound to an insulin peptide presented in an MHC class 2 molecule that's in NOD mice. It's called IAG7. And this is just looking at the specificity of this antibody. So just showing that it binds to the peptide MHC molecules that we want it to bind to. So this insulin peptide presented in a couple of different ways here. So here's our nice positive signal, and doesn't bind to irrelevant tetramers that are on the same MHC molecule. So the antibodies looked good. So we went off to generate a CAR from that antibody that Brian made using a classic second generation CAR format, because at the time we hadn't learned all that stuff about first generation CARs. And we showed that you can engineer Tregs from NOD mice. This is just showing expression of Helios and FOXP3 in our either control Tregs or our insulin B G7 specific CAR Tregs. This is just showing expression of the CAR here. And this is confirming peptide specificity. So in this experiment, we are staining our CAR Tregs with tetramers that are either presenting an irrelevant peptide, so HEL or this 2.5 hybrid peptide, so they don't bind, but they do bind to this insulin peptide. So just proving the specificity and also proving that this peptide MHC molecule will stimulate division of the CAR Tregs. So this is pulsing splenocytes with either the specific or the non-specific peptide or a positive control, showing that the CAR Tregs will proliferate in response to this peptide. So this is all with artificial peptide that we've just had synthesized. We also wanted to know if these cells would respond to native antigen. And I don't see any hardcore diabetes aficionados here in the audience, but for any of you who know it, the specificity of insulin peptides is a huge field onto itself. I'm not going to go into any of the details, but the long and the short of it is that we did find that this CAR can bind to the wild type insulin B10 to 23 sequence here. And we did this by stimulating T cells expressing the CAR either with antigen presenting cells alone or that had been pulsed with injured islets. And you can see we have proliferation here in response to the islets above the background with just APCs alone. And those data are averaged here. So this is just leading up to show that, yes, we made a CAR, it binds to this peptide MHC molecule, and it can stimulate T cells. So back to this bystander suppression question, can this CAR suppress other types of T cells? And so here we're looking at the ability of the CAR Tregs to suppress those same diabetogenic BDC T cells, looking at suppression of proliferation or the ability of these cells to make IL-2, titrating in increasing numbers of our CAR Tregs, either non-specific or specific. And you can see that they very potently suppress IL-2 production as well as proliferation in these BDC T cells. And they also suppress antigen presenting cells. So this dendritic cell activation assay here, the insulin specific CAR Tregs will suppress CD80 as well as CD86 on these APCs. So this is all in vitro. Last couple of slides just showing that they also function in vivo, and we have two different models to show the in vivo functionality. One is an immunodeficient model where we're taking immunodeficient NOD rag mice, injecting them with the BDC T cells, which I showed you in the previous data, will very rapidly cause diabetes here in black. But if you co-inject our CAR Tregs here, we basically get complete protection from diabetes, much more significant than the polyclonal Tregs that only have a partial effect. And we can dive in and we can follow these Tregs by staining them with tetramers and congeneric markers and show that the CAR Tregs are basically trafficking and getting activated in the pancreatic lymph node here. These are the tetramer positive CAR Tregs, so they're more proliferative and they're also more activated in comparison to the tetramer negative cells, suggesting that they really are trafficking to the relevant site and controlling immune responses to these BDC T cells. So this is an artificial model in a lymphopenic mouse where we're putting in defined T cells. And so the last important experiment to do was ask whether these cells actually had activity in the spontaneous model of diabetes in the NOD mice. And so in this experiment, we are taking cohorts of female NOD mice, which in the absence of any treatment, after about 30 weeks, the majority of which will get diabetes. But with a single injection of our CAR Tregs here, we can protect a significant number of these mice from getting diabetes. So I will, however, note, just like we had in the skin transplant model, it's not 100% protection in this fully immunocompetent setting. And so we have more work to do for sure in terms of, we're working on that now, layering on different types of immunosuppression, improving our CAR design, testing different first generation constructs, et cetera. So first hints of success, but still more work to do. I'm just going to skip over that. So in summary, for this third little vignette, we have successfully made a CAR that binds to a peptide MHC molecule. And this is an important proof of concept, not only for diabetes, but also for CARs in general, because the concept that you can make a CAR against a peptide MHC molecule opens up the possible antigens you could target in autoimmunity or cancer or really any disease you're trying to control, either with a CAR T cell or a CAR Treg. Expression of these peptide MHC specific CARs, people had debated a long time about whether the number of peptide MHC molecules would be enough to stimulate a CAR. There was a lot of thought that the antigen density would not be high enough, but our data suggests that it is sufficient to induce CAR activation and antigen specific proliferation. And these insulin specific CAR Tregs can prevent diabetes driven both by these pathogenic BDC T cells, I didn't show you all the data, but they reduce their engraftment, they reduce their proliferation, they reduce their cytokine production and change their ratio. And then they also work in the spontaneous model of diabetes. So I want to end with my last slide and just highlight that this is just the beginning of Treg engineering. I only talked about this one little part of it, which is trying to mess with the antigen specificity of the cells. But work in my lab and other labs around the world is in parallel pursuing ways to edit these cells, to introduce functionally advantageous molecules or remove deleterious molecules, try and move towards the use of third-party or allogeneic cells by removing potentially dangerous MHC molecules or T-cell receptors, engineering tissue homing, and then there's this whole field of synthetic cytokines, which is also very exciting and has lots of potential in Tregs as well. So tons of work to be done and lots of potential. I'm just going to end there and thank the people in my lab who did the work. So the work on the CAR signaling domains is all being done by Isaac Rosado Sanchez in the lab with tons of help from Majid Mujibian. And the work in diabetes, infectious tolerance has been done by Vivian Fung and Christine Wardell. And thanks to our collaborators, especially to Brian Fife and team at the University of Minnesota for all the work on the diabetes CARs. So I'm going to end there and look forward to discussion.
Megan, that was fantastic. I'll open the floor to any questions. Why don't you go ahead and start, and we'll let people collect the thoughts.
Get my water.
You might start by identifying who it's from if they wish to be identified.
This is Jim Bustle. Jim, go ahead.
Thank you. I was really interested in the idea of the polyclonal Tregs versus the specific Tregs. So can you give us some clue about how much specificity is required? Meaning, could you suppress almost everything if you had the A2 cells and an A2 graft, or how would that work?
So, okay, there's layers of answers to that question. So first of all, by giving specificity, what we're trying to achieve is to get the cells activated at the right place at the right time. And there's a lot of debate about what the best specificity is. For organ transplant, it seems like MHC is a good target. But then your question, I think, is once you get the cells there and you're controlling in the environment and you've got them activated, can they suppress other potentially undesirable immune responses, such as T cells that are specific for a viral infection? And that is definitely something that needs to be investigated. So far, all the people treated with non-engineered Tregs, just polyclonal Tregs, there's been no evidence of an increase in viral infections. But now that we're using these more potent cells, it is a potential side effect, which has not been investigated in basic science or in people. So it's something that we're setting up to investigate.
Thank you.
Thanks, Jim. Last hand up right now is from Jamie Scott.
Hi there. I hope you can hear me. I'm in California. Anyway, hi, Meg. This is a wonderful talk, by the way, and I wanted to ask you about this last set of experiments, and combined with the previous one too. So, do you have any evidence? I've got a couple questions here. Do you have any evidence of recruitment of new Tregs in your system? And if you do, it would be interesting to know if you're getting, quote, epitope spreading, where you know what I'm saying. So and I think that one way to look at that would be to maybe look at the PCR sequencing to see if you've got diversity there, because they're T cells, so they must be binding the T cell receptor, different from, potentially different from the CAR.
Yeah. Hi, Jamie. Thanks. It's so nice to hear your voice after all these years. Those are great questions. So we can, now that we have models that are fully immunocompetent, so the NOD model and the Black 6 transplant model, we are setting up to do exactly those experiments. So stay tuned. We hopefully in a year or so might have some data. Maybe we'll send it to you. You can help us analyze it. But I guess the hope would be, yes, that we have managed to control, that we've managed to induce new Tregs, and that those new Tregs would have multiple specificities that collectively are now controlling the immune response to a whole host of autoreactive proteins or alloreactive proteins, depending on the model.
Sweet, very sweet.
Megan, I've got some really naive questions. Anybody else in the audience before I, please?
Hi, Megan. With the second generation CAR Tregs having the intracellular co-stim already in it, do you think that would cause faster cell-cell decoupling? And then would that also cause the CTLA-4 to bind less CD80 and then strip them less?
Yeah, so in our in vitro experiments, we don't see a difference, and also that one in vivo experiment. But the CD80-86 is just a small window of what Tregs can do to APCs. And I wonder actually if by taking away the requirement for that interaction, if it may diminish the effectiveness of the Tregs in terms of reshaping APC function. So I think those data are relatively recent, and we're going back now to really look in more detail about the biology of the first versus the second generation cells, because I think overall the first generation cells will probably be better and be better at tolerance, but we'll have to see. Lynn?
Thank you so much for a great talk. You were mentioning like how the CAR T cells and CAR Tregs, like you realize that they're so different, especially in terms of CAR structure. And as far as I understand, in the case of CAR T cells, you don't really always want like the highest affinity, because that might also cause more exhaustion at the end. I was wondering if that's the case for CAR Tregs, and if there's any requirement on the actual specificity of the CARs themselves.
Yeah, so that's a question that's to be answered in the field, the question of low versus high affinity. We have a study, I didn't talk about it all today, that shows Tregs are susceptible to exhaustion, as are conventional T cells. And so it is possible that high affinity may be undesirable. We actually had a whole project to do a phage display scan of that A2 antibody and create a bunch of different affinity CARs, and we just never got around to it because of this annoying pandemic. But we need to come back to it and ask that question. So we don't know the answer. No one else. And part of the problem is there's only 3 or 4 labs in the world working on CAR Tregs. So there are a lot of questions and not a lot of hands. So if I need more people working on CAR Tregs.
Thank you, Megan, for a great talk. I have 2 questions related to about the BDC T cells. It's very interesting they can change to the FOXP3 positive. Just curious, do you also look at their function? Do they change their, like say, for example, cytokine or cytotoxicity?
Yeah, I didn't show all those data, but they have severely diminished cytokine production. So TNF and interferon is way down. I don't think we stained them for any cytotoxicity markers, but the fact that they're not killing the islets, I guess, suggests that they probably are not cytolytic or have diminished cytotoxic activity.
Yeah, sorry. And the second question is there a specific ratio, like how many Treg you have to put in to like kind of balance this reactive T cells? And if yes, how do you setting up in, say, in the human setting in the clinic? How do you interpret how many Treg you have to put into the human to kind of limit this?
Yeah, so it's totally unknown. So most of the experiments, the initial experiments in the lab were mostly feasibility, like how many cells can we make, and then we'll divide them up and put that many in. And now it's really, we have so many questions, and one of the ones we haven't got to answering yet is trying to do a dose response to see where we hit the upper and lower limits. In the clinical trials that are going on now, the cell numbers have been dictated by feasibility. How many can we make reliably from a person, and we'll put that many in. So we still, I think even in the cancer world, that question is not fully answered, the number of cells you need. So definitely needs more research.
We're a little bit past the hour now, so Megan, I'll wrap this up and say thank you again very much for joining us and giving a terrific and informative talk.
Thanks, everyone.